Hello everybody and welcome to the video on NCBI or National Center for Biotechnology Information. So as an introduction, the NCBI is a part of US National Library of Medicines, that is NLM, and it was established in 1988 under NLM, and it provides access to the biomedical and genomic information. So the NCBI, as you can see, it is a part of NLM, and this NLM is a part of National Institute of Health, that is in Maryland, USA. So it comes like a cascade, right? So the whole system, it plays a very crucial role in bioinformatics research and database management. Then let's look into the broader version, that is the International Nucleotide Sequence Database Collaboration, that is INSDC. So here it is a collaboration kind of thing between the different database systems, that is the NCBI, EMBL, EBI and ADBJ. So as we look into this, the NCBI or National Center for Biotechnology Information, it includes the GenBank, Biosample, Bioproject, BLAST, PubMed, etc. etc. And under European Bioinformatics Institute, that is EBI, we have the ENA, Biomodels, Expression Atlas, and Uniport, etc. Then under DGBJ, we have that is DNA database of Japan. We have the Reflex, CRISPR Direct, and DNA, etc. etc. Okay, so all these three databases are linked between each other, and if we get into the interface of one data bank, we can easily jump into the interface of next and next. So they are all interconnected, and it is like a collaboration. Okay, they work together. Now, when we look at the history and mission of NCBI, as we said that it was founded in 1988 under the NLM, and it aims to develop advanced information system for molecular biology, the DNA databases and informations related to DNA, nucleotide sequences, protein sequences, etc. etc. Then supports research by providing free access to vast array of biological databases. So the access is almost free. Now this is the interface of NLM or NCBI. So let's get in the interface through Google. Here we have Google. And this is the link. When we click over the link, we see all these things, right? So this is the interface. You can log in if you have the access, and we have this welcome page. Welcome to NCBI, National Center for Biotechnology Information. And here there are all data banks. Among this we can select the data bank we want. That is an option. Then over here we have, this is the NCBI home page which we are seeing. Now resource list, all resources, chemicals, bioassays, DNA and RNA, gene expression, all this you can, as you can see, you can take. Then popular resources, the PubMed, Bookshelf, PubMed, Central, BLAST. We're running a BLAST to know the sequence similarities. Then nucleotide, genome, SNP, single nucleotide polymorphism, gene, protein, PubChem, etc. Then the news and blogs page is there, and over here you can submit the or deposit the data or manuscript into the NCBI databases. Then we can download. Then we can learn, that is to help find documents and attend the class, etc. The development, analysis and research. So this is the main home page or interface of the NCBI. Now let's go back into our class. So this is the interface that we saw. So let's move to the aim of NCBI, that is to develop public databases, which is mostly free to access, and it helps in the development, research and such areas, to develop software tools to spread biomedical information. Okay. So now let's move into the NCBI data sets, that is the web interface. As we saw the interface, we can move into any other data bank or anything by single clicks. Everything is available on the interface homepage itself, and that's one thing. Then the API, the advanced programmatic access to gene, genome and viral databases. the access. Then command line tools like BLAST, etc. easy to programming, use programming access to gene, genome and virus databases. Now, when we look into the database and tools, as we already saw, it houses a series of databases. So, for example, like GenBank, a repository of nucleotide sequence. Then PubMed is for biomedical literature. Then BLAST is for sequence alignment. Then RefSequence is for curated collection of reference sequences. Then GEO, gene expression, or Omnibus for functional genomics data. Then Entras, retrieval, it's a retrieval tool, and Bankit. Then Sweekin, submission tools, as we saw the submission tools. Then specialized tools like ePCR, SpinDay, etc. etc. All these tools are there. Then when we look into the tools, we can broadly classify like NCBI into database set and tool set. So in tool set comes this data retrieval tool and BLAST and specialized tools. And in under databases, there comes the nucleotide databases, the literature databases, protein databases, expression databases, structure databases, chemical and other databases. So these are the broad classification of all the facilities that are given by the NCBI. Now let's look into the role of NCBI in bioinformatics, and the main role includes like maintenance of biological database. That is primary, that is the original submission by experimentalists, and content control by submitters. So the secondary derived database is like a build up from primary. For example, you did an experiment, you isolated bacteria and you did a sequencing of its genome, and you found the sequence ATGC sequence, and then you are submitting it into the database. Okay, so this is a primary data. Then there is something like you are designing, like you don't know, you got a sequence maybe, and maybe you are designing like a protein. That protein can be coded by a specific sequence A, B, C, D, because some kind of similar sequence was coding the similar protein in some other organism. So that is, you are just deriving, maybe this may not be accurate, it may not be correct, perfect, but we are just elucidating a possible outcome or possible structure or sequence or something like that. That is the secondary derived. Then next point is that it facilitates data storage, retrieval and analysis. That is analysis, for example, when we run a BLAST, we can see the similarity of the sequence that we got to the sequences that are already present, that already exist in the database. Then it supports sequence alignment, that is the blasting and annotations. Then provides computational tools for molecular biology research and helps in genome sequencing projects. Now let's look into the major symbols that are used. So this is a symbol that we see for nucleotide, sequence database example in GenBank and all. Then genome, then taxonomy, structures, domains, 3D domains, OMIM, then SNPs and UniSTs, GEO, PopSet, Unigen, homologous gene, homology, cancer chromosomes, and GenSat, protein, PubMed, Pub. So these are the books, and these are just major ones. There are many more. Now, how to use NCBI effectively? That is, utilize PubMed for literature reviews, and BLAST for sequence similarity alignment, and GenBank for genomic sequence data, access GEO for gene expression studies, and leverage use of for computational analysis. So the basics is that you have to know what each and everything is for. There are many commands, there are many options in the interface as we saw. But we have to actually first know what this interface is, what its application is, then only you can use it effectively, right? So applications in research and industry, drug discovery and development, genetic research and personalized medicine, agriculture, biotechnology, evolutionary and combative genomics, disease diagnosis and treatments. Here, if we elaborate one, like you have a drug, maybe you have a disease and it is related to some protein X, and this protein X is having some active site like this. So you can search in the database for some ligand that have a shape like this, maybe, so that it can fit into the active site, right? So you can list out possible ligands that can function as a potent medicine or drug, potent drug. Okay, it is not necessary that it will be the drug, but it can be a potent drug. You can go select those ligands and go for the different kind of research and use it for drug development. So such kind of applications are there. Now, future developments and conclusion. When we look, there are continuous updates in genomic databases can be done, and integration of AI and machine learning for better analysis and expanding open access tools for global research, and NCBI remains a cornerstone for modern bioinformatics research. So these are the Okay, so that's all about the NCBI. Thank you. I hope you understood. I hope you got an overview about NCBI, its different applications, and how it works. Okay, thank you.